This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 634, recorded on July 1st, 2020. Wow, that's it for June. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Madison, New Jersey, Brienne Barker.
Hi, it's great to be here. It is sunny and 71 Fahrenheit, so almost 27. Although we had a huge thunderstorm earlier today, so it's kind of nice to finally see this blue sky.
Yeah, I didn't notice that. I must have had the thunderstorm here. I'm not too far from you.
Yeah, it was a big storm for about 2 or 3 hours.
Wow. Well, I was teaching this morning, so I didn't notice. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. 87 degrees, cloudy, and what looks to be about a 50% chance of coronavirus.
In Texas, you got a 100% chance of coronavirus.
Yeah, you gotta be careful.
Well, you're not going anywhere, right?
Nope.
And from Ann Arbor, Michigan, Kathy Spindler.
Everybody here, it's 85 Fahrenheit, 29 Celsius, and it's sunny.
I think it's sunny now. Boy, Texas.
Wow, Brienne, 71 is sunny. Sounds good.
Mm-hmm.
Yeah, no, it's definitely pretty nice.
I can't see I'm home today. I'm in the basement. There's a window, You know how basement windows are, right? Do you have a basement, Kathy?
Yes.
You got windows like high up, right?
Mm-hmm.
Yeah. You can't see much.
Vincent and Brienne, how far are you from each other?
Not far enough. Just kidding. Just kidding. No, I'm just kidding. I like Brienne. She's a nice person. Let's see. Madison, New Jersey. Let's see exactly how far.
I know you've driven between your place and Drew.
Let's see. It is 13 miles.
Whoa, that's nothing.
Actually, I could drive all local roads. Yeah. Would be about a half an hour. Yeah, she's very close. Doesn't take long. I was there pre-COVID, right?
You were.
Yeah. That was a long time
When
ago.
was that? It was a long time ago.
I don't know. It just seems like, June's over. Look at this. 6 months into 2020. I think 2020 is a train wreck of a year.
Yep.
Yeah. I don't have a lot of great things to say about it.
It's really, and my course is over tomorrow. That went quickly. 24 lectures in 6 weeks.
So talk about train wrecks. I've been having this thought recently that this pandemic actually is just a warning shot. Okay. Because a lot of this has to do with the friction between science and society, and how the science doesn't care what you think. It does its own thing.
Mm-hmm.
And this is peanuts, man, compared to climate change. Okay.
I was looking at some of the really old pandemics, like before we had public health, Black Plague, like 200 million people died.
Yeah.
And they had no control because they didn't know what was going on. They were sloppy and filthy and rats everywhere and feces everywhere. Well, not bubonic plague, but you get the idea. And so, at least we got past that. At least there's some sanitation. You could sell that to the politicians. But now you can't even sell a face mask, right? Still people are not getting it. I don't understand. I found some neat pictures of face mask things on Instagram. I put in the show notes for you guys to see. You must have seen some of these, right?
I hadn't seen any. These are good.
Yeah, I'd seen the Freddie Mercury one. I really like that.
Yeah.
No mask on your face. You're a big disgrace, spreading your germs all over the place. That's really good. I thought Kathy would like that because she could sing it.
Right.
Then there's the one from The Mandalorian, which you wouldn't know unless you've seen this. It's a Star Wars TV series where the guy wears his helmet the whole time. And these Mandalorians are like warriors. And whenever they say something, they end it by saying, "This is the way." And why are you wearing that? this is the way. Never take your mask off. I love it. It's great. And then the person compares anti-mask rhetoric to anti-vaccine rhetoric with a visual. Isn't that great?
Mm-hmm.
So
Yeah.
in New York, I would say a quarter of the people walking around don't wear masks. What's it like in Austin? You don't see anybody, right?
I don't know. I don't see anybody. I mean, the only exposure I really have is my neighborhood, and everybody is so far away from everybody else, like a street's width at least, that very few people wear masks, and I think that's justifiable under the circumstances.
What about you, Kathy?
I would say about the same. When I've biked into work, I've seen some parties of students on their porches, and I'm sure they're not all in a bubble together and they're not wearing masks.
But yeah.
Oh, and the bar in East Lansing that last week had 42 associated cases now has 100.
Oh my goodness.
And you go there often, right?
No, but my brother tells me that we did eat there once. So.
And what about Madison? What's it like there?
For the most part, everybody I see is wearing masks. I'm not going to say that they are all wearing them correctly.
Right.
But most everyone I see is wearing a mask. And with your Instagram photos, there was another one that sort of went around that was funny to me, I guess, because of my age group from The Princess Bride. It's actually a quote from one of the characters who, when they ask him why he's wearing a mask, and he says, "Oh, it's just that masks are terribly comfortable. I think that everyone will be wearing them in the future."
That's right. That's the Dread Pirate Roberts, I think.
It is. And so
cool.
That's another one that's gone around quite a bit.
Wear a mask, folks. There's no reason not to.
I had a close look at the statistics on Austin. The metropolitan statistical area, 5-county area around Austin, to kind of get a grip on this today with some specific questions in mind. So the place is on fire, as I've said before. The cases are going up. And as far as I know, we are not yet maxed out in terms of the hospital capacity. But the new cases, 50% of the new cases are accounted for by age groups from 20 to 40. But there are only 25% of the hospitalizations. 50% of the hospital, the vast majority of the hospitalizations, of course, are over 40, increasing in frequency as you get older. So my interpretation of this is that the younger crowd are cooking it up and passing it off to the people who need the hospitalization.
Yeah, that's the problem, right?
Wear a mask, dude. Okay.
If not for you, for somebody else. Yeah, for some old person.
Yeah.
Who's going to get it. And, so we had our neighbor, we know them, her daughter graduated from college, and so she wanted to have a little get-together for her and her family. And I said, are people going to wear masks? And she said, no, my families are all Republicans. They refuse to wear masks. So I said, I'm not going. Can you imagine it's political now? Is that ridiculous?
That's nuts that it's political. And it's gotten, and actually the politicians are responsible for that.
Sure.
Okay. They have politicized it. And it's really too bad because because it's so visual. Okay. So that now we have this really sort of aggressive resistance to wearing a mask as a political statement. Yeah. And it's pretty, it seems to me, maybe I'm making this up, pretty, it's pronounced, let's say, in Texas. I don't know that it's exaggerated as anywhere else, but I have this, they got this slogan of come and take it, this sort of defiant, revolutionary
take my gun, right? Yeah.
attitude. Yeah.
Well, there's stories of people going in stores where it says wear a mask and they don't, and they have fights with the store owners. Right. Don't go in the store. Yeah. Enforcement is a real problem, right?
Yeah.
Yeah.
It's a real problem.
You just wish that people would respect others. Go
Yeah.
ahead, Kathy.
I was just going to say, back when, Vincent, you were talking about other pandemics and things, I have kind of a hard time keeping track of the numbers of these and keeping them in perspective. So I went to the Johns Hopkins dashboard, and there's a little over 500,000 deaths in the 6 months.
Mhm.
Compare that to the 50 million deaths that we talk about for the 1918 flu.
Yeah.
So, and, this isn't yet as long in duration as the 1918 pandemic, but we're only 1/10 of the way in death.
Yeah, I wonder how far we are, really.
Well,
is it? I mean, I don't know. I don't know the answer.
yeah. I mean, we have the problem of, deaths being not necessarily being reported, but I have to think that was probably the case in 1918 as well. That
Yes,
Well,
absolutely.
We also have the problem of really don't know how many infections there have been. Okay.
Yeah, for sure.
Even with, as we've discussed recently, even with the best testing, it could be that there are a lot of people who are functionally immune and yet don't even have an antibody response. So, if we got a lot of asymptomatic infections, and if we got a lot of people who are recovering with just an innate and a T cell response and little, if any, antibody response, we're not going to count those people. So we don't really know.
Well, all the mild infections too that never get tested.
Yeah.
Right. There are a
Right.
lot, at least 10 times more than we're testing, and maybe more. Who knows? And Barrick said that from very beginning. There are at least 10 times more infections than we're picking up. And that's why it explodes after you come out of lockdown, right? Because these people are walking around.
So what would be nice in a sort of a perverse sort of way is if there's a lot more infection than we even know about, and that we're approaching herd immunity faster than we realize.
Maybe.
But we don't know.
Well, in New York, the latest estimate is 20% of the population has been infected, which is still not close to what you need, but that would make sense in such a dense population center, right?
Right.
Mm-hmm.
Anyway, actually, we're going to start off with some non-SARS-CoV-2. Kathy found something quite interesting.
So yes, this is my brother Bill sent me the BBC link about pig flu with pandemic potential, and the primary article is a PNAS paper from China. A number of co-authors. I didn't check if it's co-first authors, but the very first author is Hong-Li Sun. The last two authors are George Gao and Jinhua Liu. And the title is Prevalent Eurasian Avian-like H1N1 Swine Influenza Virus with 2009 Pandemic Viral Genes Facilitating Human Infection. And the gist of it is that they've been doing surveillance and looking for different influenzas in swine. And there's this one called EA, where the EA stands for Eurasian Avian-like Lineage. And these have increased in their diversity since 2013. They swabbed almost 30,000 pigs, normal pigs in slaughterhouses in 10 provinces, and they got 136 samples. And they found, they typed them, and they found that this EA-H1N1 was the most prevalent subtype in the pig populations. So they took them and sequenced them and figured out that these viruses have undergone increased diversity. It's kind of a weird way to put it, but they have increased diversity since 2013. And so they're calling this a G4 EA-H1N1. And they do some comparisons with to test the zoonotic potential of these viruses. And they compare them to some G1 strains. And they find that the G4 strains preferentially bind to the human influenza sialic acid, the 2,6 linkage receptor. And so they preferentially bind, and that's a key prerequisite for infecting human cells. And then they showed that these G4 viruses efficiently replicate in 2 different kinds of human airway epithelial cells. And then they did studies in ferrets to look at infectivity and transmissibility. So they infected 3 ferrets intranasally with each virus. And let's see. So the G1 viruses or the pandemic 2009 virus only caused mild clinical signs, but these G4 EA viruses, which are now the prevalent ones, have much more severe clinical symptoms. And so these reassortants showed increased replication and pathogenicity in ferrets. They looked at the transmissibility either by direct contact or respiratory droplet transmission. And so we've talked about this in the past. They have different cage setups where they can measure direct contact or just by respiratory droplets. And bottom line is that there's compelling evidence to show that this predominant G4 reassortant virus is highly transmissible by both direct and respiratory droplets among ferrets. And then they look at whether there's antigenic cross-reactivity of these G4 viruses with extant human influenza vaccine strains. And the bad news is there is not. And then they look at what they say is an increased infection rate in humans as evidenced by increased seroprevalence. So what they do is they collected serum samples from swine workers at 15 farms and then compared them to samples from people in normal households. And the swine workers had an increased odds ratio, in other words, an increased likelihood of having positive serum to this G4 virus than the general population group. And they give numbers on that and so forth. But
if I remember correctly, wasn't it 10% of the workers had antibodies?
Yes, 10% had antibodies. You're right. And so what they're saying is that it's of concern that human infection of G4 virus could further adapt to humans and increase the risk of human pandemic. And then there's quotations in the BBC article of a lot of people saying, yeah, we can't just be paying attention to SARS-CoV-2. We need to be paying attention to these other things and continue to do surveillance on these kinds of things.
So there's evidence from what you've said. I haven't looked at the paper. From what you said, there's evidence that these viruses have infected people, right?
Yep.
They're seropositive. Yes.
But we don't, is there any evidence that they have caused disease in people or that people have transmitted it from one person to another?
No.
Okay.
No evidence of either of those. But,
that doesn't mean it hasn't happened. But
exactly. How many asymptomatic SARS-CoV-2 cases there are, people could say, I just got a little sick. That wasn't flu.
So this paper got a lot of press. And so I read it and answered some questions about it. Because there are some people who read it online who don't know a ton about viruses and said, oh my gosh, now we're going to have a second pandemic in 2020. And it's not that kind of level.
Yet.
Yes. Well, yes. But it's certainly something we should keep on.
So there's a question. What should we do? What should be done? Because this obviously could continue to infect and then maybe acquire transmissibility among humans, because obviously it's infected people, but it hasn't gone anywhere. So what should we do? Should we stockpile antivirals? Should we make a vaccine? But if that's the case, it might drift by the time it comes out, right? That's not probably...
Right. So what one of the people in the BBC article said was we should definitely have the people every 6 months who are deciding what viruses go into the vaccines be paying attention to this. But until there's evidence of human to human spread and or a human disease, I think we just have to be alert and be aware if you're asking what we should do.
Maybe they should do some sero surveys outside of China to see if it's been anywhere else.
Yeah.
Because the pigs are shipped all over the place. And I don't know if they go out of China, but China, makes more pigs than any other country. And it imports a lot too.
Yeah.
Well, this emphasizes to me how little we know about pathogenesis and transmission. You can't look at these things and say, oh, all this has to do is this or that, and be more pathogenic in
Mhm.
humans or transmissible in humans. Takes me back to the gain of function days, right? And we just don't know.
Nope.
We don't know enough to know what the pandemic potential of a virus like this is just by looking at it,
But
or how many steps away it is.
They did a lot of the right experiments, right?
Mhm.
Yeah, they did.
The human lung cell cultures, the ferret transmission, the serological studies. That's just about everything you'd want. The only thing not done is maybe look at pig farms outside of China.
Mhm.
But that's all you get from this.
And
Well, I
if
think they're doing the right thing. They're doing the surveillance.
Exactly. And so the one thing I'd want to know, if I remember, it was 2013 to 2018 or 2011 to 2018,
Yeah.
where they did the surveillance, and this virus became predominant in 2016. So, I want to know what's happened in the 2 years since then.
Right.
Um,
2011 to 2018. Right.
so I'd like to know what's happened since then to continue with the surveillance, just like Rich said.
I think we need to make transgenic pigs resistant to influenza viruses and use those for meat.
Mm.
Then the virus would, of course, change to be able to infect them. I don't think you could make sialic acid negative in pigs. I think you need sialic acids. So,
Yeah,
so
just
much.
to reemphasize what Brianne said, it was 10% overall seropositive, but in the age group 18 to 35, it was 20% seropositive. Of course, we're talking small samples, 9 out of 44. But
These are all swine workers too, right?
yes.
I noticed this was submitted for review on December 9th, 2019.
Mhm.
I guess they got it out just before they started getting busy over there
Mhm.
at the CDC. Wow. 3 first co-authors,
Okay, good.
co-first authors. That's pretty interesting.
Yeah.
Wouldn't that be horrible if this emerged later this year? Wow.
Oh,
Yeah.
God. Don't even go
Fortunately,
there.
masks and hand washing and physical distancing would all act against it as well.
Yeah, if
Mhm.
you wore masks for the rest of the year, this would not emerge. So please wear masks.
Mhm.
If you don't like it for SARS-CoV-2 because of influenza, it would help in the fall too. Oh,
That's
boy.
the other meme I like. If you think a mask is uncomfortable, wait till you're on a ventilator.
Is that a
Oh,
meme? That's
yeah.
something that's out there?
Yeah.
I think they are right. It's like anti-vax, same idea. I'm
Yeah.
not doing that, even though
This has become a personal, individual rights thing.
it says
and
here
a
on
political
this
thing.
on this Instagram, it says the premise is the same: refusal to do the bare minimum to protect large portions of the population based entirely on poor data and fake science. Healthcare isn't an opinion.
Actually, this I had a conversation with the guy who runs the Brighton Collaboration Group that I work with, who was actually an EIS officer, Epidemiologic Intelligence Service, quite a while ago. Worked for CDC forever. I
Is that
don't know quite what
Merchulinski? Is that Merchulinski?
no, this is Bob Chen. I've talked about him before. Robert
yeah.
Chen. He
Yes.
actually was a co-author on that Lancet commentary that was published along with the Wakefield article, taking it down point by point and predicting what a disaster it was going to be. Okay. Nobody paid any attention to that. Okay. At any rate, he's a public health expert, and he says public health is a combination of science and politics by definition. And if the politics goes wrong, public health goes wrong.
Okay.
I'd never thought of it that way, but that's right.
It's funny. I had never thought of it that way either. And then I just thought about our public health major we designed in the courses we have to take. And science and political science.
Okay. Now, I thought this article came out yesterday in Clinical Omics. VaxArt's COVID-19 oral vaccine candidate gets a boost from Operation Warp Speed. And I thought we'd talk about it because of the oral part. And, Kathy knows about this. Right, Kathy?
Okay.
So this is an adenovirus,
okay.
vector, right? So how does that work?
So the AD4 and the AD7 vaccine that's given to military recruits is an oral vaccine. So it's encapsulated and it's a live attenuated vaccine.
So it's an enteric coated capsule, right? So it makes it
correct
through your stomach and into your intestine.
I guess, yeah.
I think that was
think
my
so.
understanding
And
of how
then-
that worked.
Then the virus
So
reproduces.
it infects, winds up releasing the virus in your intestine, so it infects the intestinal mucosa.
All right. And then that protects you against the respiratory adenovirus infection, correct?
Yes.
And how did they figure that out to begin with? Did they know? I mean, why would you even think to do that? I'm wondering.
So,
I don't
Brianne,
know.
this goes
Yes.
back to the mucosal immunity is sort of
Exactly.
in your gut is connected with your mucosal immunity elsewhere. Is that right?
Yeah. So both of those are sites where you have a lot of mucosal immunity, where you make a lot of the antibody type IgA. And so perhaps if you first encounter a virus in a mucosal site, then you're going to make more of a mucosal biased type of response.
Hey, for the uninitiated, let's see if we can define mucosal or
Ooh.
mucosum. So this all makes sense.
Okay. So the mucosa are a set of organs that are in contact with the outside world and have sort of specialized structures to help them deal with the problem of pathogens in the being with the outside world. So they usually have a layer of mucus protecting cells. They have some special immune responses and things like that.
So we're talking about gut, mouth, nose,
Mhm.
vagina,
Mm-hmm.
gut, including rectum.
Yep.
Okay, I
You
think
can
of
also
them
think
as,
about lungs.
I
and you
think
can
of it as slimy skin.
Yeah, you can sometimes think about the conjunctiva too.
And so somehow inducing, so there is an immune response that is specific to the mucosum, right? This IgA, which a different kind of antibody that
Yes,
shows
so
up
that's
in
a
the
different
mucosum.
kind of antibody that is really present in the mucosa.
And somehow these things are all connected so that stimulating a mucosal response in your gut winds up helping you in your respiratory tract. Is that
That's
right?
the idea, but what's Kathy
Yeah.
have to say?
Well, I was just going to, since Dixon isn't here, I was just going to put it, the gut bone's connected to the lung bone.
Yeah, so there's been this idea in creating vaccines for a while about whether with a mucosal pathogen, you need to use a mucosal delivery and think of a way to push the immune response towards a mucosal response. So whether that's needed and whether that's possible has sort of been debated among vaccinologists for a while.
And that's
It
the
looks like
idea
it
behind
worked here.
FluMist. That's the idea behind FluMist, right? That's a nasal live attenuated influenza virus. And theoretically, it's good because you're delivering it to the normal route of infection.
Exactly. That's the idea behind FluMist. And there you're, going to the respiratory tract. And so you want to kind of teach your adaptive immune cells that they should head back to the respiratory tract.
And so this is kind of a perversion of this because, Kathy, my understanding of the military adenovirus vaccine is that the virus, it's the regular wild type virus that's delivered in this capsule. Would you say 4 and 7?
Yeah, I was thinking it was attenuated, but maybe you're right. Maybe
I think it's the wild type virus.
This is
That's
5.
delivered to the gut, but
This is type
if
5.
it infects the gut, there's it's no symptom, but you do get an immune response.
It's type 5.
This vaccine is type 5.
Yeah, this
Right.
particular one that we're talking about, yeah.
But the military one.
It's 4-7,
Right.
yeah.
The principle is copped from the military
Yeah.
vaccine.
So they said they have made multiple candidates encoding various combinations of the spike protein and one or more targeted internal proteins for evaluation. On April 30th, the company disclosed it had obtained positive preclinical results for its candidates with, quote, several, unquote, generating immune responses in all animals tested after a single dose, which, by the way, doesn't mean a darn thing. You don't need to just generate immune responses. It has to be protective, but they're not showing that. And then they said a final candidate was selected for manufacturing, and we don't know what's in that, but it must be spike plus something else, or
It would be nice if it was plus something else.
they don't tell us. And I think all they did was stick it in animals and get an immune response, and they wanted to start a phase 1 by the end of the summer.
So, this sort of makes me a little uncomfortable with the immunology, although the fact that it's based on the vaccine that's given in the military makes me feel like there must be some good science behind exactly what's going on.
You're being
but
kind.
I always think about immune responses in the gut as being sort of their own special thing.
Yeah.
And that the immune system in the gut is really unique because it has to be able to tolerate food without making immune responses to food. And so there's sort of this unique immune response going on there. And so when they say they made immune responses,
Where?
Right, where and what type, and
They don't say.
are they
Yeah.
protective? All sort of come in my head because I'm worried about this being a GI vaccine. But like I said, because it was working in with the military, that's some evidence that there is a way this could work.
They don't know if this would protect. They don't know if this would work for SARS-like viruses. That's the bottom line, and they're have to. I don't know how they can do an efficacy in humans without some animal data, right? Maybe they go into phase 1 without animal data.
Well, that's the whole idea behind all of this warp speed stuff is, I don't want to say cutting corners, but at least compressing.
Yeah. So what is warp speed, Rich?
Oh, you're asking me?
Operation Warp Speed. It is Trump's commitment to deliver 300 million doses of protective vaccines by January 2021. Protective vaccine. Well, we're going to see what that means in a moment because we have the FDA document, but
They say it's a partnership among components of the Department of Health and Human Services, the Centers for Disease Control, the Food and Drug Administration, the National Institutes of Health, BARDA, which we've talked about before, the Department of Defense, and down here we've got a link to the site if you want to put it in the show notes. In terms of leadership, HHS Secretary Alex Azar, Defense Secretary Mark Esper, Dr. Monsef Slaoui, Slaoui, designated as Chief Advisor, and General Gustav F. Perna, nominated Chief Operating Officer. Okay, so it's a government operation. 300 million doses in January. Wow.
So this is only selected vaccine candidates also, right?
Yes.
Right? Now, you know what I like about this is that it's a tablet that you can take orally really easy. Most of the other ones are injected. I think that's great. I hope it works. But I'm really worried about all these spikes in the egg basket, It really, since we discussed that last week, let me tell you why. Remember the Dengvaxia, where they took yellow fever vaccine and they stuck in the structural proteins of dengue types 1, 2, 3, and 4, right? And 1, 3, and 4 generated protective antibodies. They all generated neutralizing antibodies, but 2 did not generate protection. And the idea was you needed some of the dengue non-structural proteins to give you a nice T-cell response. So even if you make neutralizing antibodies in that dengue story, it's not enough to protect. Right. And that's a justification for putting spike in. You can make neutralizing antibodies against it. How do you know that's enough? You don't. So I think this is a huge gamble.
Well, they do say that some of their candidates, this company says that some of their candidates have other antigens than just spike in them. We don't know what the details are in this.
I hope so.
I hope they've chosen well. Kathy, Ad5. Now, in my recollection, adenoviruses can cause a spectrum of different diseases, from conjunctivitis to respiratory infections to GI infections, and some can even do more than one thing. Do you know what Ad5 ordinarily does? I'm wondering basically how respiratory. Yeah. Does Ad5 do any give you any GI symptoms? Do you know?
Not that I know of. Not that.
Of course, it's an attenuated Ad5.
Right.
So Ad5 bothers me because we've all got. I mean, the seroprevalence to Ad5 is really high, right?
It's pretty high. I've just been looking that up while you were talking. The seroprevalence for Ad5 and its kissing cousin Ad2 are are pretty high. I mean, AD2 and AD5, are really quite similar. And for AD2, it looks like it's 45% had moderate and 27% had high titers for AD2, and 27 and 9% for AD5. And this is a population in China. And then there's another paper from Dan Baruch's lab looking at AD5 versus 26 and 35, which are two of the favored AD vaccines. So, yeah, the seroprevalence is always a concern.
So the other another adenovirus vectored vaccine that's on the list of 5 candidates that Operation Warp Speed has selected out is an AD26 vector from Johnson & Johnson / Janssen Pharmaceuticals.
Yep.
Right.
And I think the seroprevalence for that is very much lower.
Correct.
So, which is one of the reasons to go for that. I wouldn't be, well.
But if I remember correctly from those papers from Dan that Kathy's mentioning, AD5 has the best transgene expression. And so you have sort of this trade-off of if you use one of the others, you have less pre-existing immunity, but you also don't express whatever you've put in as well.
Yep.
And I agree with Vincent. The real magic of this thing, the real attractiveness of this thing is the pill.
Yeah, it's great.
That'd be awesome. I wonder if you could deliver AD26 orally. I wonder if that would work.
I don't see why not.
Yeah. One of the others on this list of 5 is the formerly Chadox-1, right, which has now been renamed. I kind of like Chadox,
Yeah.
Yeah.
Flows off the tongue. That's University of Oxford. So that's chimp adenovirus, right? And then
Somebody said to me, you probably can't have chimp in the name of anything.
Yeah, that's true. That's
Successful.
true. Oh, no. Yes, that's a good point. I like that. Then we have...
and the new name of it is AZD 1222,
Yeah,
which does
right.
not roll off the tongue.
and then we have BNT 162 by Pfizer and BioNTech, which is another spike mRNA vaccine. And then we have mRNA 1273 from Moderna and an unnamed vaccine from Merck.
Which I looked it up, and where did I put that?
You said it's VSV Delta G vectored.
Huh.
Yep. So
All
same concept, at least. I don't know if it's exactly the same vector, but same concept as the Ebola, the VSV vectored Ebola vaccine.
All spike based. Okay. We'll see if that's a good idea or not, right?
And the other thing, the question that came up in my mind is that a few weeks ago, we talked about another collaboration called ACTIV, the acronym ACTIV. And I'm loading up the link here slowly. But this was in a Science article that was, here it is, co-authored by Lawrence Corey, John Mascola, Anthony Fauci, and Francis Collins, that describes a collaboration between both government and private industry in an effort to produce a vaccine. And I was wondering whether these guys are talking to each other and whether they're the same or different, what was going on. And Kathy had some insight into that.
Yeah, because I asked my former student Marty about it, and he said that he thinks Warp Speed has more to do with manufacturing, and ACTIV is a network of clinical trial sites.
So hopefully these guys are all talking to each other.
Right. And if you want to get BARDA funding, you have to use active clinical development because they want to make the clinical endpoints consistent and comparable. And one of these 5 turned down BARDA funding. I think it was Merck. I can't remember which one turned it down. But Vincent, back on your thing about too many eggs in the spike basket or too many spikes in the basket, whatever it is, this Vaxart thing, they don't tell you in the end what it is.
Yeah.
But it could have an additional protein in it
Yeah,
Right.
because they were trying that.
I think
So-
that would be good. Yeah, they don't tell you, but the more apparently you can make neutralizing antibodies against the N protein also, so that would be a good one to have in. I don't know what else would be good as well. So that's better than nothing.
And I am aware from talking to others that one can make coronavirus virus-like particles
Yeah.
with that are comprised of a number of different structural proteins, because there's, in addition to spike, there's matrix, and I forget what other proteins involved. So something that a vaccine that approached something like a virus-like particle or had all the appropriate antigens in it would seem a logical approach.
Yeah.
We'll see.
I
It
agree.
could be that a good, robust antibody response to spike is enough. I hope so.
Could be, but the fact that so many patients don't seem to have such is telling us maybe spike antibodies are not the whole story. I mean, they started these vaccine candidates long before we had any serological results from patients, and I'll bet now that those conversations in the rooms where they decided what to do would be different. So we'll see.
I'll just test all of their immune responses in my eBay EliSpot machine.
There you go.
That's right. Did you order it?
I did not order it.
How's the GoFundMe campaign going?
It got a couple comments on Twitter.
There you go.
So yesterday, the Wall Street Journal had an article called FDA to issue guidance on COVID-19 vaccine. And then this morning, and so they said basically that today the FDA was going to send out a document saying how to do these vaccines. And today, a guy from the FDA, Andrew Farb, who is Chief Medical Officer in the Office of Cardiovascular Devices, sent TWiV the document. Now, maybe Andrew listens, or maybe, I don't know. Well, I'm glad that you sent this because I wouldn't have found it. It's a very long, comprehensive document called Development and Licensure of Vaccines to Prevent COVID-19 Guidance for Industry. This is what you're going to have to do, folks, if you want to make a vaccine. And, my overall, I want to go through some points because I think they're really interesting. But overall, it seems to me that they are going to be tough. I mean, yesterday, the article said they will not approve anything that is not at least 51% effective at preventing infection or disease. 51%. And I'm sure Kathy remembers what the efficacy of the first Salk vaccine was as announced in Rackham Hall in 1955. Do you remember that, Kathy?
I don't remember the efficacy.
It was like 54%.
Okay.
And that was considered pretty marginal, but it was approved because, why? Because parents wanted it to protect their kids.
There wasn't anything else. Yeah.
And I remember in that press conference, Salk, one of the things he said, I'm already working on a better vaccine. And Basil O'Connor, who had run the fundraising for him, got so mad at him. He said, why did you say that? It's a fine vaccine already. Why did you say that? You're going to make a better one. Well, he got mad. People got mad at him for many reasons also. Mm-hmm. But anyway, so 50% is like not a big deal. But I guess that's what we need. Anyway, so here's some points I want to mention. First of all, they say vaccine development may be accelerated based on knowledge gained from similar products manufactured with the same well-characterized platform technology to the extent legally and scientifically permissible. And they say so that means if you have a platform that works, we may be able to do things faster, which I think is okay. But then they say, we recommend you talk to us early before you start something to get feedback on whether we think you can do that. And I mean, conversations, communication, I think that's really important. So they go through, general considerations, non-clinical data, preclinical data. The purpose of non-clinical studies is to define immunogenicity and safety characteristics. And the extent of how much you need to support the first-in-human trial depends on the vaccine construct, the data you have, and data from related vaccines. Okay. So you got to have some, immunogenicity that's all it is. It's not protection.
Right.
So they're not saying you need to show protection at all.
Well, but that makes sense because if they're doing this in an animal model, you can't necessarily show protection.
Well, you could. Non-human primates can be done.
Yeah.
They've done some. You can do protection in mice. You can do protection in ferrets, but apparently they're not requiring that. So for phase 1 anyway. So
right.
And they say talk to us about this. They have lots of more information. Oh, here's one for Brianne. Studies should include an evaluation, and this is for these preclinical studies. Evaluation of humoral, cellular, and functional immune responses as appropriate to each of the included antigens.
Nice.
I don't like the as appropriate, though. Why put that qualifier in? That allows someone to say we don't need it. Functional is like a neutralization assay, right?
Yeah, functional is a neutralization assay or, looking at cytokine production of the cells.
They say
I guess
functional activity should be evaluated in neutralization assays using either wild-type virus or pseudovirus.
Yes.
Studies in animal models. Oh, then there's a section on potential for vaccine-associated enhanced respiratory disease. And they say, we don't know if there's a link, but you should look at it in your animal models. We consider that important. They want to have high neutralizing antibody titers, and these will allow vaccine candidates with high neutralizing antibody titers may be allowed to proceed to first in human without first completing post-vaccination challenge studies in appropriate animal models. Okay, so there it is. If you show neutralizing antibodies, you could go to phase 1 without a protection study in your animal. And then there's a section on clinical trials. Listen to this. Understanding of SARS-CoV-2 immunology, and specifically vaccine immune responses that might predict protection against infection, is currently limited and evolving. That's what we say all the time, right? We don't know what's protective. So the goal of development programs should be to pursue traditional approval via direct evidence of vaccine efficacy.
In protecting humans.
Protecting humans.
Efficacy as defined by protecting humans from disease. Okay, that's the critical thing. Just an antibody response or something isn't enough because we don't know if that's a correlate of protection, really.
No, it's very important because you can imagine some pressure to just license something that induces an antibody response. Then the trial populations. This is very interesting. Older adult participants over 55 may be enrolled in early phase studies as long as they do not have medical comorbidities. This is common. You got to use healthy. But, this is important because if you just test your vaccine in 30 to 40-year-olds, you can't give it to Rich and me.
Right. That's the case for the flu mist vaccine, right?
Yeah.
It wasn't tested above a certain age group, and so that age group can't get it.
To generate sufficient data to meet an approval standard, late-phase clinical trials to demonstrate efficacy will likely need to enroll many thousands of participants, including many with medical comorbidities, for trials seeking to assess protection against severe disease. Thousands.
This is where I have trouble with the notion that we're going to have 300 million doses of vaccine available in January.
Absolutely.
Oh, yeah.
Okay.
Absolutely.
Because if you need a phase 3 trial that's got like 30,000 people in it, that's just a number that I have heard. I don't see. It's July, right?
Yeah.
I don't see how we're going to have results from that between now and January. Now, it could be that they, as we said, they're going to compress stuff. It could be that anticipating success with some vaccine, maybe you can do, if you started like today, maybe you can get a trial done by January, and maybe you can anticipate success and have a lot of doses available by then. But boy, that seems mighty compressed to me.
Well, so if you look in that trial design section, there's another bullet point, which from an immunology point of view makes perfect sense, but that bears on what Rich is saying, that says follow-up of study participants for COVID-19 outcomes, in particular for severe COVID-19 disease manifestations, should continue as long as feasible, ideally at least 1 to 2 years, to assess duration of protection and potential for vaccine-associated ERD as immune responses to the vaccine wane. And I think ERD is enhancement.
Yeah, it is.
Yeah.
Well, that's after,
Enhanced respiratory disease.
that's after licensure, so that's typical, right?
Yeah, I think it's important for people to understand that just once something is licensed, in a way, the clinical trials aren't over because the companies are obligated to follow up with the people that they vaccinate to make sure that on that sort of large population scale, there aren't adverse effects that crop up.
Vincent, I just want to check. I think you were saying that efficacy has to be 51%. It says 50%. And it could make a lot of difference if we're talking that
50 or more, right?
The vaccine was only 54. So at least 50%.
Yeah. Another point I thought was interesting. They say, although establishing vaccine safety and efficacy in naive individuals is critical, safety and outcomes in individuals with prior infection, which might have been asymptomatic, is also important to examine because pre-vaccine screening for prior infection is unlikely to occur in practice with the deployment of licensed vaccines. Okay, so it's interesting because maybe having a prior infection would influence the outcome, and you want to know that because you're not going to check people before you vaccinate them when you finally do vaccinate millions of people. So if there's an issue, you have to learn, right?
Or even people that weren't tested for the antigen, but were tested for the antibody, had really seems like had to have had the disease, but their antibody test is negative. They're going to get the vaccine.
Yeah.
But maybe they did have it and their antibody test was flawed. So even if they had been tested, they're going to get it.
They say we encourage the use of diverse populations so that the vaccine can be safe for everyone, right? All kinds of, and they include racial and ethnic minorities and pregnant women and others as well. And they say a randomized control trial with a 1-to-1 randomization between vaccine and placebo is the most efficient study design. And they say there are others, but talk to us if you want to do something else. And then there's this whole section on efficacy. How do you measure efficacy, right? They say either lab confirmed or disease or virus infection is an acceptable primary endpoint for a vaccine efficacy trial. So you could look for a virus preventing infection or actually preventing disease. And they have a whole list of things you could monitor for disease. Obviously the virus is PCR, but other things you can do. And they say because it's possible that a vaccine might be much more effective in preventing severe versus mild disease, sponsors should consider powering efficacy trials for formal hypothesis testing on a severe COVID-19 endpoint. Very interesting.
Very interesting.
At least 50%. There it is. You're right, Kathy. Then finally, safety considerations. The general safety evaluation of COVID-19 vaccines, including the size of the safety database to support licensure, should be no different than for other preventive vaccines for infectious diseases. And you got to have at least 3,000 study participants vaccinated with the dosing regimen intended for licensure. Anyway, that's all I wanted to talk about here.
I really want to thank Andy for sending us in.
Yeah, I think
this is great.
This is really tells me that FDA is not going to be pushed around,
right?
Which I think is important. Which was one of the worries of Paul Offit, which we talked about a few episodes ago in his article or his interview. So thank you. Yeah, thanks, Andy. Okay, now we have something slightly different. So this some time ago, Dave Hall, who teaches immunology at the University of Utrecht, he sent in a letter and said, could my students send questions in? As part of the course, they'll get credit for these questions. And so I said, yeah, why not? And so he sent in a bunch. This class was taught this year on Zoom. It's called Signaling and Techniques in Immunology. Taught by Paul Koffer, Kristen Denzer, Jorg van Loosdrecht, and Dave Hall. So let's go through some of these, okay? And rapid fire answers. Yaneke. I was wondering, what is the likelihood that SARS-CoV-2 mutates its S protein, leading to the particle binding to a different receptor?
No.
Not likely.
Not likely.
Nope.
Rich.
Robin, I have a question for the podcast. It is known that SARS and MERS were able to infect dendritic cells and alter interferon expression and maybe other stuff. The similarities between SARS-CoV-2 and SARS and MERS assumes that SARS-CoV-2 also infects and modulates dendritic cells. Dendritic cells are essential for vaccine-inducing immune response. Do you think the dendritic cell modulating properties of coronaviruses have to be taken into account for vaccine development?
Yes,
I'm going to turf that one to Brianne.
So my answer is, so I have two answers. One, yes, that would be fantastic. And I think that studying these things is really an important thing to be studying. One thing that we can also imagine is if we're not using the full virus in a vaccine, we're just using one or two proteins, they may not have the same interferon alteration ability that the real virus does.
But of course, all the vaccines in development probably are not looking at any of this. They're just popping in spike and sticking it in animals.
Except for a couple of vaccines that are actually dendritic cell-based vaccines that are on the preclinical list. Okay, that's a platform technology we haven't discussed before.
Okay. Kathy.
Monica writes, with regard to SARS-CoV-2, I'm guessing that the best vaccine candidate would be one that can provide neutralizing antibody as well as cellular immunity. If generated and it provides high enough titers of neutralizing antibodies that give sufficient protection of disease, would we then be able to induce sterilizing immunity? That is, to get rid of or neutralize all virus that gets to the upper respiratory system. So if we can get an immune response that generates high titers and generates cellular immune responses, would it be sterilizing?
I'm interested to know the answer to that.
Yeah, I mean, basically, from what we know about the human coronaviruses, that does not seem like a probable outcome. Okay. On the other hand, as we've discussed before, a vaccine doesn't necessarily have to mimic natural immunity, and could be that you could make a vaccine that gave you a really robust humoral response. I'm making this up. Okay. And I'm not an immunologist. So
I support it.
Okay. A robust humoral response that maybe would even give you sterilizing immunity. Who knows? We'll have to see.
If you remember that Chadox, when it was tested in non-human primates, it protected against lower tract disease, but not upper tract replication. So, that's the one now in phase 1 or 1-2, whatever. So I think it's not likely, but we'll see.
It just occurred to me that one of the important things that these are questions coming from students, right?
They are. Mm-hmm.
I like it when a student can, regardless of whether the idea is, very far off the wall, if they can formulate a question that makes sense using all of the appropriate vocabulary, I'm impressed. Mm-hmm. And this qualifies.
Yeah.
Okay.
Yeah,
that's a good question. Good question.
Absolutely.
I agree. Brianne.
Joanne says, what a nice opportunity. Thank you for that. I had a question regarding the observed lymphopenia in COVID-19 patients. How is SARS-CoV-2 able to induce lymphopenia? Or is lymphopenia more a result of innate immune evasion by SARS-CoV-2?
Well, it's a good question. I found a paper on it where they document the lymphopenia, and they have a couple of ideas.
Let's define lymphopenia. Make sure I'm on the same page.
Reduced numbers of lymphocytes.
Okay.
Yes.
So, yeah, the one thing that comes to me because of my simple mind is the virus could infect them and kill them. But I don't think there's any good evidence for that yet, frankly. Right, Brianne?
No, there's not. There are a couple papers on it, but I they require more data before I will support them.
How common or exaggerated is this effect? Is this a common thing?
Yeah, that's a good question. Let's look at
See
the
what they
paper.
say.
This study was done on how many patients? Blah, blah, blah. They did it in 5 patients, 5 death cases. So I don't know how common it is. I don't, from listening to Daniel, not sure how common it is.
Right. This is not something that's really prominent on my radar.
Yeah.
I mean, I think more broadly, sort of immune dysfunction might be more prominent, but I don't know if the specifics of lymphopenia are common.
And there are other possibilities in the paper, like it's cytokine imbalance and et cetera. So,
right. There's another paper. It's the one that I sent Vincent and Brianne, also to Stephanie and Cindy, building on this idea that coronavirus might be depleting immune cells and in some ways behaving a little bit like HIV, causing this depletion. So that was another paper that I thought, maybe you guys should cover that on immune.
So the answer to the question is we don't know.
Don't know.
Yes, the answer is we don't know. And I know that when Jonathan Udell was here, he mentioned something about potentially reduction in B cells leading to this problem with antibody positivity. And that's stuck with me ever since.
All right. Next is Sam. Since SARS-CoV-2 is expected to infect such a large part of the population for a long time, are we going to have dramatically distinct serotypes? Comparisons with flu? Might it evolve into a serotype that is attenuated and less severe? Do you think it will remain with us in a more tolerable serotype? I don't know. But looking at the common cold coronaviruses, they've been around a long time. They don't change their serotype. You build population immunity, and that causes a less severe disease outcome. And Ralph Barrick suggested that might happen with this virus as well. But for none of the coronas, correct me if I'm wrong, we don't see multiple serotypes, right?
No.
This is a one-trick pony.
Right.
And that's what I do is I look at what's happened with similar viruses in the past. It could always change, but that's my take.
I'm thinking this idea of antigenic drift, which is, I think, what is on people's minds with a vague familiarity with flu. If you look across virus families, that's not very common.
No, it's not.
Most of the viruses I know, look at measles. There's one serotype, and it's been that way forever. Mumps, same way. Poliovirus, we have 3 types that bother people, and it's remained that way for a long time. Flu is really a pretty unusual story in this regard.
Hey, smallpox, one serotype, right?
Yep. Well, actually, that's interesting because there was another. I don't know if you would call it it's not a serotype. There was a variant. Okay, that actually cross-reacted with smallpox. That was showing up at the time, around the time the vaccine came into more global use. It was called elastrum, which was a virus that had a significantly lowered mortality rate. All right. And I have often wondered whether over time that might have displaced smallpox.
Interesting.
We'll never know, but.
Quite interesting. All right, Rich, you're next. Sarah.
Let's see, Sarah?
Yeah.
Thank you for your email about information about the podcast. This morning, Dr. Fouché talked about computational software that's being used to map the previously circulating influenza strains. This appeared to be rather linear, indicating that the evolution and arising of new influenza strains is more predictable than previously presumed. I was wondering if such techniques could be used in order to say something about the evolution of coronaviruses and maybe even predict the potential arising of future coronaviruses. Are these your notes here, Vincent?
Yeah. And Kathy. But
go for it.
Well,
I'm gonna turf
basically
this.
it's the same thing. Could there be drift? And first of all, that there was some presentation implying that maybe drift could be predicted, but I would think that what was presented was retrospective and showing you how things went along. I don't think there's any ability to predict future mutations for flu or, for that matter, for coronaviruses. And so this kind of comes to the same idea: is are coronaviruses behaving like influenza? And I don't think there's drift. I don't think it's predictable. So I don't think we're going to be able to predict things for coronaviruses.
Kathy, you're next.
Kunal, thank you for this opportunity. My question is, with the worldwide rush to develop a vaccine or antibody therapeutics for this disease, there's also talk of the risk of antibody-dependent enhancement associated, leading to unintended consequences among patients of SARS-CoV-2. How would a researcher take this into account, and would this change any parameters for testing viable candidates for treatment or vaccines? So to review for the listeners, what this student is asking about is, if a person had SARS-CoV-2 and then got it again or got the vaccine, as we were just discussing, could they potentially get a worse disease, as we know happens with dengue virus? And we're going to punt this one to Brianne.
Yay! So the idea is that antibodies that are not neutralizing are able to allow the virus to enter a different type of cell. So that's sort of what is usually thought to be happening in the dengue immune enhancement situation. And so I think that what we would want to do is look at people with different antibody levels, and perhaps look at some of those patients with pre-existing infections or previous infection, and see if getting a vaccine leads to potentially some issues with them, or really look at what type of disease we see in someone who's been vaccinated, who then gets COVID. And so we sort of have to look at those patients after they've been vaccinated to see what happens with them.
So there's actually two types of disease enhancement that are described. One is the antibody-dependent disease enhancement that you described, that is where dengue is the good model, and another is I don't know if there's a role of antibodies necessarily, but it's respiratory, what do we call it? Enhanced respiratory disease, like they saw with the RSV, respiratory syncytial virus. Okay. We had a paper that described this that I'm trying to come up with.
Yeah, I remember that too, but
I will look for it when I'll find it. At any rate, there's been, people are concerned about this. There have been some evidence in animal models that this might happen. Ultimately, you got to wait to just keep that on the radar as you're evaluating the safety features of the vaccines, right?
Yeah, and the FDA document says you should pay attention to this and figure out animal models to check it.
Right.
Yeah.
You try and you do the best you can with animal models, but you're really, I don't think, going to know whether this is an issue until a vaccine is deployed in humans, and that's going to be a very important part of the safety considerations.
Absolutely.
And of course, the precedent is the feline infectious peritonitis, where there is ADE demonstrated in cats. So that's the only corona, I believe, for which you can see that. So it can't happen with a coronavirus. Brianne, can you take the next one?
Sure. Saskia writes, polysaccharide protein conjugate vaccines have been very effective in providing protection against bacteria. She gives a citation. And recently, the extensive glycan coat of the S protein of SARS-CoV-2 has been simulated and is suggested to lock and load the S protein for ACE binding. And she gives another citation. However, current vaccines that are in development are not using this vaccine approach. Due to the extensive glycosylation and potentially important role of the S protein glycans, and due to the identification of non-receptor binding domain targeting antibodies, she gives a citation, how would such a polysaccharide protein conjugate vaccine compare to current vaccines that are in development in terms of type of immunity induced and clinical response? Please do not sugarcoat, pun intended, the answer. So most of the polysaccharide conjugate vaccines I am aware of are against bacteria. As is mentioned here, I can't think of any examples with viruses. This is certainly something people should be looking at, because I do not know enough about the glycan coat on S. But certainly, I think experiments should be done to look at whether or not that glycosylation impacts antibodies.
But of course, we're not. We're just making vaccines. I mean, presumably,
eggs in the spike basket.
So if you look, this paper is quite cool. They modeled the sugar coat. The spike is extensively sugar coated, and they say here the epitopes. Some of them are shielded, some of them are not. So you could imagine maybe trying to target antibodies to an unshielded epitope. But whatever is in these vaccines is going to be sugar coated the way it is in the virus, because it's being made in a cell. So the genetically encoded vaccines, the mRNA and the DNA and the vectored vaccines. So I guess, it's going to be sugar-coated in the same way. But whether that makes a difference, who knows? Yeah, I think it's really worth looking at. But that's fundamental research that we don't have time for, right? Sadly. Maybe in the longer run. Laura G. had a question very similar to the one about drift of SARS-CoV-2. Is it like influenza virus? So we've answered that. So that brings me to Sarah, who wants to know. She said, I read there's evidence that the virus spreads through exosomes and that this can induce an immune cell response. How exactly does this happen? I looked very hard. I couldn't find this evidence that it spreads through exosomes. Exosomes, of course, are little membranous vesicles that come off out of the cell, and I believe they're released from intracellular vesicles, right? And they can carry proteins and RNA to other cells. And so, picornaviruses, which are naked icosahedral viruses, a fraction of the time they are released in cells in exosomes, and they have a little membrane. Hepatitis A virus, for example, does that quite a bit. The idea is that it's thought to shield the particle from antibody response. So it happens with viruses. I don't know if it happens with SARS-CoV-2. Does anybody know?
No.
Okay.
Seen anything?
And the last one is from Rich's.
Okay, so
You've been looking around. You want to?
I've been tuned. I've almost got it, but it's okay. Where am I up to?
Dave.
The last
H.
one, Dave. Dave H.
Dave H. The world assumes a cure is possible for SARS-CoV-2. Okay. Correct me if I'm wrong, but to date, the most successful antivirals are too slow. Whose stay in the body is quite long years. Viruses, HIV, etc. I'm a little confused about what's going on.
So he's saying that the most successful antivirals are to slow viruses.
Like HIV. Okay.
Yeah.
However, the research community has tried and failed to make inhibitors to a variety of viral proteins of fast viruses, for example, picornaviral protease that substantially alter the course of viral disease. I suppose the only exception would be Tamiflu. And SARS-CoV-2, indeed all coronaviruses, seems to stimulate only a weak antibody response that does not last long. No one seems to know why. Are there other viruses where the immune response is better studied that might serve as a similar template to understand SARS-CoV-2? This looks like kind of
two questions,
more like a comment and a question, or two questions. One is about antivirals, and there's always a problem with antivirals with an acute virus infection of being able to deliver the antiviral early enough in the infection to actually have an effect.
I don't think it's a matter of not being able to make them. It's what you said. You can't diagnose them in time to make a difference.
Right.
We have plenty of picorna antivirals that work in the lab, work in animals, but nobody wants to make them because there are no diagnostics. Tamiflu is an unusual one, and because there are rapid diagnostics for flu, right?
And the stimulating a weak antibody response that doesn't last long. Now, this is on the fringe of my understanding, but what comes to mind is respiratory syncytial virus. I don't know that it's a weak antibody response, but I do know that it wanes, and you get reinfected with RSV time and again during your lifetime. Subsequent the reinfections being milder. So immunity in that case seems to wane, but I don't think anybody knows why in that case.
So I bring you back to John Udell, where we had a discussion and we concluded that if a virus causes a viremia, it makes a gang-busting, lifelong immunity, like measles, whereas the respiratory viruses that stay away from the blood, influenza, coronaviruses, RS, don't. At least that's a correlation. I understand it's not causation, but that's an interesting, and even Brianne liked that. I don't know.
I did think it was really interesting, and I started asking a lot of my friends who work on different respiratory pathogens, does your pathogen cause viremia?
And polio does, and it makes a gang-busting, lifelong immune response. So, and that might be part of it, but what's going on? Who knows? Okay.
Found the article.
You found the article. What is it?
Yep. Barney Graham.
Barney Graham, right. He's working on SARS-CoV-2 vaccines, by the way.
Yes, he's part of the NIH arm of the Moderna vaccine, if I'm not mistaken.
Yes, that's correct.
And the article is in Science, and it's titled "Rapid COVID-19 Vaccine Development."
Oh, by him, right? Yeah, yeah.
And in the body of the article, we've discussed this before, I believe. He has a really nice description of the variant forms of enhanced disease and what's known about what causes.
That's right. That's really a good article. I remember. Thank you, Rich. And thank you to all the students for sending your questions.
Yes, very much so. And good luck to you.
Kathy?
I really liked the way that they were able to take information sort of across different disciplines they've learned.
Mm-hmm.
I love it when my students do that. That's a sign of really having learned stuff well.
Mm-hmm.
Kathy, you're next. This is the regular email now, right?
Dan writes, Dear TWiV, I've been a fly on the wall of your conversation since the end of the world began earlier this year. Thanks for what you do. I was so excited to be offered the opportunity to return some expertise in the form of mere counting. I'm a software engineer living in Portland, Oregon, and counting up the length of all your episodes is definitely something I can help with. All the episodes total to 3,598,179 seconds, or about 999 hours, 29 minutes, and 39 seconds. Assuming you could manage 16 hours a day of nonstop TWiV listening, this would turn into about 62 and a half days. One way to look at this is that the total body of TWiV content is about the same length as the lockdown for this pandemic has been at this point for people like me who are more than happy to stay home. On a personal note, I was hoping the number would come out to be something quite different, so I could put some joke in here about being happy to prove Alan wrong, as he seems like the kind of guy that needs to be proven wrong now and again. Takes one to know one. Oh, well, not this time. He was pretty dang close. Perhaps the next episode should contain a 1,000-hour celebration. Dan. So, and then he gives a PS, a note on his methods. This comes from the duration tag in the RSS feed that you provide on the site, the resolution of which is 1 second. Thus, there may be a small amount of rounding error, approximately 0.018% maximum error. The feed contains 650 episodes, which I assume covers any special or unnumbered episodes, as TWiV is currently at 632 as of this count. So I think then we've already passed the time for our 1,000-hour celebration.
I think we have.
We're about an hour and a half into this. So now we're at 1,001 hours. And it's just amazing that Alan's tossed off number of about 1,000 hours was so
something.
Close.
I have to say, many people sent in this. All right, besides, what is this? Who is this here? Besides Dan. So thank you all for sending in your.
I appreciate the fact that our listeners have a sense of humor.
Mm-hmm.
Yeah, and I like when you say, "What is this?" They respond with answers, and that's just great. Brianne, you're next.
Mike writes, Dear Twivers, in a recent episode, Dixon thought that a reader response complimenting the show sounded like it could be a haiku. I tried to capture the spirit of that response in the following haiku, entitled An Ode to TWiV. COVID-19 clouds can only be dispelled by the sunlight of TWiV.
Ooh,
Nice.
Nice.
that's
Great.
very nice.
That is great.
That's a really
That's
good
like
one.
our third
That-
haiku, right?
Yeah,
That's a good
Yeah.
one.
this is a keeper. Not that the others aren't as well. This is good.
Thank you, Mike. This is really good.
It's really great.
Maybe COVID. The Sunlight of TWiV, episode title.
Yeah, I'm just putting that in.
Good. Very good.
I'm on it.
People like the last title a lot, by the way.
Which was that?
Countering a miasma of anti-think. I actually shortened it from what we had decided on. I thought that worked well. People liked the word miasma.
Are you worried that people were going to get mad at us for that?
No. Usually there's neutrality in the titles. So when you get response, that means people like it. You know, the rule of thumb on the internet, if one person responds, that means 1,000 people like it. But they never answer. They're just lurking, right? George writes, I'm a long-term listener and fan of TWiV based in Wales, weather 16C61F. Hey, 16 is 61. Look at that, Kathy.
There you
I know.
go.
He's got it.
And Drizzly, I work as an OBGYN, but spent a year in the middle of my medical degree studying immunology and virology at the University of Bristol, at which time one of my lecturers kindly put me onto TWiV. I've been hooked ever since. I am enrolled in the Oxford University SARS-CoV-2 ChAdOx vaccine trial, phase 2-3, and received the vaccine a week ago. Naturally, I have read up on all the minutiae of the ChAdOx platform, and it's all the more exciting to follow the trial when I have skin in the game. Kind of literally. For my arm of the trial, there's a 50% chance that I received the experimental vaccine and 50% that I received the control, MenACWY vaccine. To be eligible to participate, I was antibody tested at the outset to ensure that my SARS-CoV-2 serology was negative. In passing, the epidemiologist administering my vaccine mentioned that the experimental vaccine would make me test positive for future serological tests. I don't know if that's true. Maybe not, right?
They hope it will.
They hope it will. That's it,
Well,
exactly. You don't know if it's going to work in every person. Not every vaccine does, right?
That's the idea of a trial, right?
Yeah.
We'll find out.
Yeah, the National Health Service here in Wales is now rolling out routine antibody testing for staff members like me. This is primarily for the benefit of EPI and public health, but those tests that are informed of their result, I hope you can help me with my dilemma. Do I accept one of these tests, which would effectively unblind me? While possible that I could have ended up in the control and also had asymptomatic infection since vaccination, right? One of these tests would almost certainly tell me whether I had the experimental vaccine or the control. As a further twist, the majority of trial participants are healthcare workers and so will be offered this serological testing like me. I suspect most of them will opt to be tested. Should I act like them in order to make our behavior more homogeneous, consistent? Part of me feels I have a duty to expose myself to lots of others for the benefit of science, irrespective of whether I receive the experimental vaccine or control. The trial data will be stronger if I expose myself. What stops me acting on this is the knowledge that I could be putting my patients and other contacts at risk. Finally, in response to what is now emerging as virology's greatest question, do farts spread coronavirus? I can report that some urology doctors at my hospital have been insisting on using comprehensive PPE before performing rectal exams on any patient. Well, I think it is definitely better to be excessively cautious. While we still have much to learn about SARS-CoV-2, I'm sure that any fart transmission event would be so significant as to be instantly published as a case report. This is yet to happen. Thanks for everything you do and for keeping my enthusiasm for virology lifelong. The fart question, oh my gosh. I was on a Zoom with the committee at Columbia today, and someone said, I was laughing like crazy at the 4-year-old's fart. Oh my gosh. All right, so I think you could have had an asymptomatic infection and you would be seropositive. You don't know if the vaccine is giving you seropositivity or not. If you turn out to be seropositive, you have no idea whether it's the vaccine or a natural infection, unless they are also testing you for antibodies against the vector, Chaddox, chimpanzee, whatever it is, adenovirus, which I'm sure they're not doing, right?
So what I think is that he should find out if there's a way to get his result, but not look at it until the trial is over. I don't think he should change his behavior based on what he thinks he is antibody-wise. And I think that's true for everybody. I don't think that people who have tested positive for the antibody should go out and be reckless and not wear masks and all that kind of thing. So if there's no way to get the results without opening them, he should inquire if there's a way to do that, I think.
Yeah, my question was going to be about whether his behavior would change with different outcomes here, because really he wants to, not change his behavior to help make sure the trial results are the best they can be.
Right. And as he said, if lots of people in the trial are also getting these tests, it could indeed skew the results for the reasons that he's saying. And so it seems like this should be made much more of an issue rather than him just writing to TWiV.
Yeah, I was wondering whether
that up to the vaccine trial people or to the antibody serological testing folks.
Right. I think that's a question for the people running the trial.
But I think those are all good points, but I don't think that the result is going to tell him whether he was previously infected or is seropositive from the vaccine. I don't think.
But it could subtly change his behavior.
Oh, yeah.
And,
okay.
And I don't think that should be a part of the vaccine trial.
Dr. George Winter, by the way, is at Wrexham Mail-Or Hospital. And he wrote, don't sweat over the pronunciation, but I think I got it.
Who knows? It's Welsh. That's like the one we had last week.
It's not. You don't think it would be Mail-Or? Yeah, probably not.
It could be anything.
But we're not sweating it.
All right, Rich, you're next.
Jen writes, hello from Melbourne.
Melbourne, that's right. You got it.
I got it?
Yeah.
I want to go to Melbourne sometime.
I was there a few years
ago. There's
nice.
A great mystery show called My Life is Murder, starring Lucy Lawless. They've only had one season with 10 episodes. I hope they make another one.
Isn't she the one who was in some Superwoman thingies, Lucy Lawless?
Yes, she was Xena.
All right.
Was it?
I think that's right.
Warrior
Princess.
Okay.
Princess.
Got it.
At any rate, she's terrific in this. But Melbourne is prominently featured. The photography is just terrific. Makes me want to go. Sorry about the digression there. It is 11C and raining outside. Perfect. She scratches out social.
Oh, that might be me. That might be.
Oh, is that right?
I've gone through and edited a lot of these.
Okay.
It's Kathy. I knew it was
Perfect
Kathy.
physical distancing weather. Firstly, thank you for TWiV. I'm only a recent convert to your diehard fan base, but I've enjoyed and truly learned much from Professor Raccaniello's lectures for a few years. As a biomed public health undergrad, I'm obviously an expert in my field. Wink, wink. But seriously, I've been keeping up with the pandemic since December, and as bad as it's been so far, I've been dreading the day we see indications of the pandemic spreading to the developing world, especially Africa. We're all aware that these regions face certain challenges with respect to healthcare resources and infrastructure, fragile economies, etc., where there is poverty, there is ill health, and there are significant discrepancies in life expectancy. Add to this the displacement of over 6.6 million refugees in sub-Saharan countries. However, I'm interested to know your opinions on age demographics across Africa. Looking at the median ages by country across the world, Europe has some of the highest median ages. Italy, Germany, Monaco, also Japan. These range between 45 to 48 years. Countries with the lowest, Niger, Mali, Uganda, Angola, Zambia, theirs range between 15.4 and 16.5 years. Median age for the entire continent of Africa is only 19.7 years. Half the population of Africa is under 19.7 years of age. I know a lot of issues make predictions and comparisons hard, but do you think this could have any implications on the impact of COVID-19 mortality in Africa? Or am I being too reductive? On that note, in a recent TWiV episode, one of your listeners quoted Jonathan Swift, to whom is attributable my all-time favorite and ever poignant quote, "Falsehood flies and truth comes limping after it." So that when men come to be undeceived, it is too late. The jest is over, and the tale hath had its effect. Like a man who hath thought of a good repartee when the discourse is changed, or the company parted, or like a physician who hath found out an infallible medicine after the patient is dead. Ain't it the truth? Wow. You know, it takes a crisis, unfortunately, to bring people around, and often it's too late. That's why I brought up climate change at the top of the show. Mm-hmm. At any rate, yeah, this is an interesting point. I think what she's implying is with the younger age, and given that SARS-CoV-2 seems to cause a lesser disease in younger age, might the impact or even overall infection fatality rate in Africa be lower? Certainly that's a logical conclusion, but of course all these other demographic issues play into that. So who knows how that will balance out. Interesting thought.
Issues of sort of other parts of their general health because of poverty and because of other things are going to be balancing out what's going on with age. Sure. So I think it's hard to say exactly what to see, but I agree that I sometimes worry when I think about what may be happening in some parts of the world where we're not looking quite as closely.
Different disease profiles as well. I mean, although it's not a respiratory disease, malaria comes to mind. Yeah, because there's a lot of malaria in Africa, and who knows what sort of impact that might have on how you react to COVID, how you react to SARS-CoV-2.
And other parasitic, other protozoan parasites, all kinds of things. Yeah, that's a great point. Read the rest, Rich.
Thank you for all you do, which I think may be more than you know. Please be safe, and kindest regards, Jen. P.S. Stay angry.
All right, we will. Maybe more than we know, probably. I guess. That's really cool. I like that. Yeah. Kathy, you're next.
Wayne writes, "Hello, Twivers. Another quick note and question from Wakayama, Japan. I just finished listening to the podcast that Dr. Vincent did in Singapore on mosquitoes. While walking my dog on this very hot 28°C rainy morning, I was cogitating on the idea of, if the SARS-2 is in the blood, as was mentioned in a previous podcast, my 76-year-old memory fails me on which one it was." Don't worry, all of our memories would fail us on that. But I think Dr. Griffin mentioned it. Could the virus be transmitted via a mosquito injection of anticoagulant from an infected person? Love your great wisdom and humor. By the way, Dr. Vincent, I live in Bozeman, Montana, as well as in Japan, and now there are 213 infected folks in Bozeman. Numerous out-of-state visitors come to Bozeman for fly fishing and to visit Yellowstone National Park. Early in the pandemic, out-of-state visitors were banned from Montana by Governor Bullock. Again, thanks for your great and informative podcast, A Fresh Wind Clearing the Fog of Ignorance. Wayne.
Ooh, there's another show title.
I think Wayne was he had written something that Dixon said sounded like a haiku, and that's how we got the haiku.
Mm-hmm. Wayne, thank you for listening to the non-COVID episodes.
Mm-hmm. Yeah.
So I think the evidence for any reasonable concentration of SARS-CoV-2 in the blood is slim.
Yeah.
We've talked about this before. And by the way, Vincent, I wasn't on the show, but you brought up that paper that purported to show infection of endothelial cells in patients, in pathology samples with SARS-CoV-2. And I totally agree with your analysis of that paper. they had arrows pointing to these spherical things,
Yeah.
okay, with no clue,
Yeah.
really, that was SARS-CoV-2. So I didn't buy that at all. So I'm not at all convinced that there's any significant viremia or that really impacts on disease. And so following on that, I wouldn't worry about airborne or arthropod transmission.
Yeah, Vincent Munster said the levels of virus in the blood are very low. And, West Nile virus has a viremic stage, yet it cannot be transmitted from human to human by mosquito because the virus titer is too low. So I think
most of the insect-borne viruses actually have a replication phase in the insect.
Yes, right.
So that you get some amplification in the insect. Now, there are a couple of examples of transmission that is strictly mechanical. The only one I really know of is myxoma virus, but that's not the predominant form. And there's certainly no evidence that this thing replicates in an insect vector.
Exactly.
And it's hard for me to think of two more culturally disparate locations than Bozeman and Japan.
Oh, by the way.
How do you deal with that?
So this reminds me. So I had a conversation with David Quammen 2 weeks ago, and he said, we only had 3 cases here. So that's why I mentioned that on the podcast, but apparently there have been more. But that paper with the endothelial cell, Rich, it was 2 human cases where they took these pictures and who knows what particle. There was no assay for replication. And they conclude, these are like medical reports, this clearly shows virus replication in the end. I mean, this is the problem with SARS-CoV-2 and the pandemic. You have all this stuff where people are so assertive and there's no data for it and nobody is monitoring. The journals are not monitoring it. This is like Lancet or something.
Yeah.
Right? Why aren't the editors or the reviewers saying something? Not sending it. It just drives me crazy. It's driving me crazy, and it happens every day. There's a paper published where the conclusions are not justified by the data.
And it seemed to me that in that paper they could have done more to try and nail down whether those samples had virus in them, and there must be immunofluorescence. Assays or whatever.
Of course, yeah, definitely. Oh, well, Brienne, I think you're next, right?
Yes. JP writes, Dear TWiVians, I write to you from Salt Lake City, where it is 78 Fahrenheit and sunny. Our humidity is resting at 32%, which is no small blessing in our wildfire-prone state. I'm currently finishing up my PhD, though not in a science-based field, but rather in creative writing. I am but a humble poet. I found my way to TWiV via my partner Cecilia, whose PhD is in virology. Of course, our respective fields aren't so different. We strive to make the invisible seen and the ineffable defined.
Ooh,
I like that.
Another show title. That's invisible, seen, and ineffable defined.
Awesome. As a scholar and lover of literature, I was elated to hear Daniel in episode 632 quote from Dickens' A Tale of Two Cities. Later in the episode, you mention a time-lapse graphic that compares COVID-related deaths to more familiar causes of death, thereby making the dangers of COVID more real to the general public. It occurred to me that these abilities to concretize and analogize are 2 of literature's greatest assets. Perhaps you've heard the somewhat apocryphal tale that Shakespeare composed King Lear while in quarantine, which likely influenced the play's bleak existentialist tone. And maybe you've read Daniel Defoe's A Journal of the Plague Year. Chances are no, though you're not familiar with Curie, a brilliant book by the brilliant poet Ellen Bryant Voigt, which imagines scenes from the 1918 influenza pandemic. Here, and he gives a link, are 8 sonnets from Curie as they appear in the literary journal Blackbird. I hope you enjoy them, and I'd love to hear what you think. I was wondering, too, do you have any pandemic-themed recommendations for me? Thank you all so much for keeping us informed and amused in equal measure. #stay grumpy.
So I need to make a reading list.
Yeah. You haven't been?
No. And I only have one suggestion of a book that I have not read. Niles keeps telling me I need to read The Great Influenza.
It's okay.
It's okay.
Don't bother.
You've read it?
Yeah, don't bother. Years ago. It's okay. I mean, the social parts are okay, where he talks about the impact on everyday life. But then he wanders into science, and he tells us that influenza virus converts its RNA to DNA and integrates into the host cell.
Uh-oh.
And nobody picked that up? Seriously? I'm sorry. I yelled a bit there. Sorry.
Well, you have
go
been
ahead.
cautioned to stay grumpy.
You have. One of the lines in one of these sonnets is, in the drills, they keep us 20 feet apart on account of sickness in the camp.
That's great. Yeah, I'm trying to look for exposure-related things. That's a good one. Death ripening in him like a boil. How about that? Can death ripen in you? I guess.
It's Kyrie, in case anybody is wondering. It's a part of the Mass.
I'm looking at this because it's pretty cool. I have to admit, I don't read poetry very often. I just read crappy journal articles, apparently. Maybe I should read more poetry. Soldiers trickled home to study peace. Oh, that's cool. Isn't that cool? Soldiers trickled home to study peace. Ah, that's nice. Thank you so much. There is a good book on the yellow fever outbreak. A yellow fever outbreak. What the heck is the name of it? Let me search here. Hold on. Yellow fever.
Are there any good books about Lassa or early Ebola?
Oh, Fever by John Fuller.
Okay.
That's the one that made me become a virologist. Just that, nothing else.
Okay.
So Wayne, who we just read from Japan and Bozeman, he sent this in 12 hours ago. It's called The American Plague. He said this would be great. The untold story of yellow fever, the epidemic that shaped our history. So that could be good too.
And of course, there's Spillover
By
David Quammen.
Yeah, I really like Spillover.
As background for this whole thing.
It's more like crossover infections, but yeah, that's good too. There you go.
Don't do Spillover as an audiobook. The reader mispronounces too many words. It would drive you crazy. Well, as a poet, you might not recognize the mispronunciation.
Yeah, but you don't want to learn the mispronunciation.
Yeah, you don't want to learn them wrong.
All right. Can we do, you think we could do one more round?
I can do another 2-3 hours.
Yeah.
Yeah.
All right. It's my turn, I guess, right?
Mm-hmm.
Yeah. Mona writes, Dear TWiV, my error was in my letter you read in TWiV 631. I failed to include context for why I wrote what I wrote. We figured it out, actually. In a recent TWiV, you discussed the mutation at codon 614 as it was in an alarmist preprint some months ago and was found lacking. That's great. Found lacking. So true, Mona. Oh, found lacking. I like that a lot. Right? Evidence found lacking. You discussed how many mutations had been found in the U.S., and Vincent said there were probably not many. I was trying to find the answer, and this is just info. Next strain looks at thousands of sequences from all over the world, but they have or had on that day 113 from the U.S., of which about 2/3 had the original G and 1/3 have the changed amino acid. Right, and gives the link. Maybe I'm being helpful, but maybe not. You're helpful. Of course you are. Here's another. In 631, you discuss slaughterhouses. Besides beef, pork, chickens, there are others like tuna, which is cleaned, involving many workers outside the U.S. We have a friend involved in cleaning and packing tuna in Manta, Ecuador, where there are many canneries. In his factory, they are taking many precautions, spacing face masks and shields, sometimes plexiglass separations. They have few problems, but he said the other factories which don't take precautions are having more COVID. By the way, COVID is rampant in Ecuador. So it doesn't have to be that way in a slaughterhouse, right?
Thank you, Mona.
Slaughterhouse
Yes, thank you.
19. You should write a book, Slaughterhouse 19.
Slaughterhouse. Okay.
It's a kind of a rack and yellow crappy joke.
Okay,
right?
fine. It's good.
Which,
good.
Okay, Rich, you're next.
Bjorn writes, Dear TWiV team, love the in-depth, long-form discussion. That's us, long form. I've learned a lot. I have a skating question. I am a former Olympic speed skater. Holy cow. And currently completing my PhD in neuroscience in the Netherlands. I love it. My research has me stationed in skating rinks for extended periods of time. Wow.
Why is that?
I don't know.
Is it so boring that he wants to go
Figure it out.
skating?
No. His research. He's
Yeah, I got
research in neuroscience. He's in skating rink.
Okay.
So something about speed skaters' brains or even peripheral nervous system. Who knows? August 1st, the big indoor 400-meter skating rink in the speed skate crazy Netherlands is partially opening back up. At first glance, speed skating seems like a relatively safe sport on a big 400-meter ice track, but you'd think physical distancing wouldn't be an issue. But how bad is it to have 300 skaters breathing heavily in a chilled indoor environment, skating at 30 kilometers an hour in tight packs of 5 to 20 individuals? Aren't skaters essentially going to be crop dusting each other with corona? Another show title. This is rife with them. Or do I see this wrong? Thanks a lot for the great information and discussion. Doesn't sound like a good idea to me.
Yeah, right away when you say tight packs, physical distancing doesn't seem to be possible. And I think we have a PNAS paper that was put in our Slack channel that we haven't discussed yet that describes that physical distancing is not enough. You need to be wearing masks as well.
And a chilled indoor environment doesn't sound like something with very many air exchanges.
Right.
Yeah. The only difference between this and a chorus, it seems to me, is that you're moving, but probably not fast enough.
Right. and also just as a point of interest, when we want to keep our viruses alive, we chill them. We put them in the refrigerator or we put them in the freezer. So yeah, the colder, the better.
You mean infectious, right?
Infectious. I just was correcting myself as I repeated it. Yeah.
Wait, what was I going to say? Oh, crap. Now I forgot. Just because I had to correct Kathy, now I forgot what I was going to.
And you didn't have to. I was going to self-correct.
Well, I didn't do it for you, but rather for the listeners who are going to write in.
I know.
I think this is a problem, but we don't have a controlled study to prove that.
No.
However, if they're going to open up, you're going to know if these people they should be tested. I would say test them all frequently.
Yeah.
And see if it's a problem.
Good idea. Do the experiment. You can double your PhD in neuroscience up with a master's in epidemiology or something.
Yeah.
I just love the crop dusting. That's great.
Yeah.
All right, Kathy.
Rich writes, "Hi, TWiVers again. My friend is a teacher at a high school in Europe. At their school, the children are required to wear masks when in the playground, in the corridors, and in the classrooms when away from their seat. But once seated, they can take them off. The classes are 50% capacity and have one child per desk, 2 meters apart. Would it be wiser to have the kids wear the masks the whole time instead, even when seated, remembering that they'll be spending about 1 hour in that classroom together with their peers and teacher? The restaurant infections that we've all seen in the paper does highlight that being at a different table does not mean that you are 100% safe, especially if you have a fan blowing air around. So a classroom with 2 meter distancing where kids spend an hour together doesn't seem that dissimilar. We're playing a numbers game, and every effort we take reduces the probabilities of infection by various amounts. So masks protect us and others a bit, distance protect us and others a bit, et cetera, all adding up. Here, the kids are wearing masks much of the time anyway, so it's not adding something new or adding cost or time. It's just doing something more for more of the time. Obviously, you could make exceptions for language classes where seeing lips moving is absolutely valuable. My friend points out the kids wouldn't like it, but hey, they're already wearing the masks when moving about, so they'll get over it. And if the school has an outbreak, that would be bad, as the kids' and teachers' health is at stake, and also the kids' education and grades, as an outbreak could close a whole school for weeks. A fundamental point that I so often encounter that needs to be raised is that we should do the best we can for as much of the time as we can. We can never get to 100%. I'm sure this is a named logical fallacy, but I can't think of what it would be. I so often hear, we can't do it all of the time, so why bother? Or it's only 50% effective, so why bother, et cetera, et cetera. We should highlight all these measures that are available to us, especially where it's continuing what we're doing, with no cost, no extra effort, such that we can all do as much as we can to the best that we can, and it all adds up. We all get distracted and focus too narrowly on stuff, so we probably need constant reminding of this. Loving the in-depth, detailed coverage and teachings you're giving us. Cheers again, Rich. He's a Brit living in Bali still, where it's 30 degrees Celsius most of the time, but dry season is coming. It has our sweaty, wet season contributed to our strangely low numbers out here? The lack of testing only explains part of it, it seems. So I think that you're right, Rich. I think that the kids should wear the masks all the time. Another factor being that if you're taking the masks off, are you taking them off and putting them in a paper bag and sanitizing your hands immediately? And then when you put the mask on again, are you sanitizing your hands first and putting the mask on and then sanitizing your hands again? I bet you the kids are not doing that. And they'd probably rather just wear the masks all the time. But those are the things you're really supposed to do when you're donning and doffing masks, or else you're getting whatever virus might have been trapped on either side of the mask on your hands and then spreading it.
Yeah. That's a good point. That's a good point.
I think his thinking is exactly right. Every little bit helps.
Yeah.
Yep, I agree too. And I think the logical fallacy, if it does not have a name, should be the it's better than nothing principle.
Right.
I think that's great. I think that's a great point. I mean, this raises the bigger question, should schools reopen in the fall? And from what I'm hearing, teachers are very unhappy with teaching online. They're just not effective. The kids aren't paying attention. It seems imperative to get them in class, but you have to be careful. You have to wear masks and you have to test them, but they're not going to test. I see so many excuses for not testing. So I got an email from Cornell. President of Cornell said, we're opening. Why? Because the kids are going to come to Ithaca anyway. We've done surveys and they're going to hang out if they don't go to school. So we might as well bring them into class and have them wear a face mask, et cetera. But we're not going to test because we can't. We don't have the facilities. And to which I say, what have you been doing the last 3 months? Why didn't you ramp up your testing capability? Why didn't you buy a bunch of PCR machines and train people to do the test? That's what we should have been doing. So to say I can't do it is just falling short. And I think schools need to do this somehow. Figure out how to get your kids tested. But I think I'm thinking more and more we should go back to school. What's happening at Drew, Brianne? Do you know?
Yeah. So we have, we're going to be on campus for 10 weeks. So we're having a staggered move-in where the first 3 weeks are virtual so that we can have people moving in over a staggered time. Then 10 weeks on campus with social distancing, with limits to classrooms, with wearing masks all the time. And then we've canceled fall break. And everyone's going to move out at Thanksgiving. And then there'll be 1 week of classes and finals virtually after Thanksgiving.
I think face masks and distancing and testing is an essential part of getting back to school. Testing is key. And people are making excuses why they can't do it. And it drives me nuts.
Yes, I was in a meeting this morning where something was said about the testing that we were getting ready to do and getting the equipment needed for that testing. And I kind of wanted to be like, can I have it in my lab afterwards?
One more.
By the way, Rich is in Bali and I want to go to Bali.
Go ahead.
Me too.
Go ahead.
There's an article that I didn't read yet I guess it was online yesterday, but it's in today's paper copy of the New York Times, why the American Academy of Pediatrics is calling for having students physically in the classrooms. So there's more reasons than just the problems with the disease. There's other downstream effects.
Sure. So, all right, one more. Brianne.
Richard writes, please note this chart of folks who were infected on an airplane with SARS and their seat locations. And he sends us a link. Flying unprotected by PPE does appear risky. My question is, would it make sense to fly protected by an adult diaper as well as other PPE? As I am 67 years old and have a new grandchild living in France, current temperature 57 Fahrenheit in Chamonix, France. I hope your answer is not, it depends. Thank you for your informative discussions. And I think answering mail is a great part of the podcast, Richard.
Oh, I'm glad you like it. That's what we started to do at the very beginning. This is an interesting picture. It's what I've always been saying about the spread is mostly local in the airplane. They have this index case, right? And then most of the cases are around where that person is seated, right? And there are a few outliers, but there always are. So here's my advice to you, Richard. Get a seat away from the infected person.
I did read something that said that sitting on the window edges was better.
I read that as well.
Yes, that's right. Because the people are moving around in the aisle, and you're sitting near the aisle, you're close. So you go to the window, you sanitize the wall with some gel or some wipes or ethanol wipes, right? Because the previous person may have sneezed all over it. You wear a face mask the whole time. I think it can work. However, does anyone know do airlines require face masks? Do you know this?
I do not know.
No, I don't know.
I think
I
they
have
should
seen
require
announcements recently that airplanes are going to start flying full. Some airlines have been flying with the middle seat empty, and some airlines have now said, no more. We're going to fill them up.
Okay. Airlines say everybody on board must wear a mask.
Good.
But this is New York Times Travel. I'm clicking through. By the way, this image of the plane is from a March 2003 flight. So this is SARS-1.
Yes.
And they, of course, were not wearing face masks, most likely.
Right. Okay. Airlines say everybody on board must wear a mask. So why aren't they? Companies are promoting new rules, but when it comes to enforcement, they are not following through. And the date of that was June 4th.
Yeah.
Enforcement's really a problem.
Especially on airplanes. They don't want to rub you the wrong way.
Right.
I mean, what are they going to do if someone takes off their mask halfway through the flight?
Not going to do anything. They do
Flight
not.
attendants have been told not to confront passengers who opt not to follow them.
Yeah, they shouldn't let them board. Of course, what if they take them off then, right?
Right. Earlier, when we were talking about people not wearing them correctly, I was in for my annual exam with my primary care doc and was in the waiting area, and everybody had masks, but one guy had his pulled down below his nose.
Yep, that happens too. I see a lot of that.
This question here, would it make sense to fly protected by an adult diaper as well as other PPE?
What, on the face?
Well, this is
so that you don't have to get up and go to the restroom during a long transatlantic flight.
No, I don't. If you're wearing a mask and wash your hands, I think you're good. I don't want you peeing in your diaper, man. Come on.
Yeah.
Well, I don't.
I mean, I can see
astronauts do it.
Yeah, it's true. So it doesn't. All right.
Yeah.
You know, we have to live in the world.
All right. Okay? I'm sorry. My apologies. I know that people wear them and they do pee in them, and I got it. But if you want to do that, I don't see that it would help, frankly, because aren't they limiting the number of people who can go to the bathroom? You can't queue up and all that stuff, but you're going to still walk by people. What a good question. It's up to you. If you feel comfortable wearing a diaper, go for it. I wouldn't.
I don't know that the John on the airplane is necessarily. I'm making this up, but I don't know. I wouldn't think necessarily that it's any more hazardous than anywhere else on the airplane.
Okay. Well, it's just passing
the hazard. That the biggest hazard is being in proximity to people who are infected.
Yeah, well, walking by them, isn't that enough? If you could walk to the bathroom,
exactly.
But supposedly, if you walk by somebody briefly, it's not going to be as likely as if you sit next to them for at least 15 minutes.
That's right.
And you wear your mask and you wash your hands.
As a matter of fact, maybe what you ought to do is get on the plane and go sit in the bathroom for the whole flight.
Well, you could get a seat near the bathroom, which I always avoid, but you could make a point to be right next to it. Right? Interesting question. Interesting times.
Yeah.
And I really appreciate your questions, all of you. They're to the one, they are just stimulating and really, really thought-provoking.
So many things I've never thought about before.
Yeah. Okay, that's TWiV 634. We're past 1,000 hours of programming.
Mm-hmm.
2 months, it seems like nothing.
Yeah.
After all these
I
years.
just
16 hours
really-
a day.
But yeah,
So yeah, those of us who've listened to the entire catalog have spent 2 months of our lives listening.
But if you did an episode a day, which is more reasonable, then it would be like a couple of years, right? 1,000 episodes, it would be 3 years roughly, right?
650 episodes. Yeah.
Anyway, thank you all for
2
the
years.
calculations. It's microbe.tv/TWiV is where you can find the letters and links to articles. Send your questions and comments to TWiV@microbe.tv. And if you like what we are doing, you could consider supporting us, microbe.tv/contribute. Brianne Barker is at Drew University. Thank you, Brianne. Oh, and also, of course, BioProf Barker on the Twitter. Thanks, Brianne.
Thanks. It was great to be here.
Rich Condit's an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
And I am Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.